Now, let's welcome Professor Collins for his talk on Palaeoproteomics. Matthew, all yours.
Thank you. Yes. What I will try and do is talk a little bit about how I see this area fitting into archaeology. So I've called it Palaeoproteomics, past, present, and future. And what I want to do in the very first few slides is just say a little bit about how I see what proteomics is and how we do it. And then what I want to do is actually think about how, from an archaeological perspective, we can then use the new opportunities that paleoproteomics has to offer. So my first slide, I'm going to give you my definition of what paleoproteomics is. And basically, when proteins are long sequences of amino acids, the amino acids are the building blocks of proteins. And so these amino acids exist and can be characterized in isolation. We can take a sample and we can measure the proportion of the 20 different amino acids within it. And that's been done since about 1950 on ancient samples. What paleoproteomics is doing is something different. And that's actually trying to get the information out about the order in which those samples are linked together. So we're trying to actually look at the way, the sequence between each of those amino acids. And so we are kind of, I think, all familiar with DNA, and many of you will be familiar with the way in which DNA encodes proteins. So sequences of DNA, and we have these things called codons, these three base pairs. And what they allow us to do is to read the DNA sequence and convert those three base pairs of DNA into one amino acid. So, and this is happening at vast speed in your cells at this moment in time. You are producing huge quantities of protein from your DNA, and the DNA exists in your body in tiny, tiny amounts. And you're actually making large amounts, 20-fold more protein than DNA in the body. And the way I like to think about it is, in archaeology, work in DNA has been incredibly successful. DNA is rewriting archaeological stories now, particularly about migration of people. Proteins are different because when we read a genome, we read about the whole history of the individual. And when I was a child, I was always impressed by this man that would come to the house and they would try and sell you an encyclopedia. This was a thing called the Encyclopedia Britannica. And it was a book which purported to include almost all of the important information that a family would need, so the children would need growing up. So all the information in the world was in this incredible encyclopedia. And I always wanted my father to buy us one. He never would, because you bought the first copy and then you had to buy so many other volumes to get all the information. And it was actually very expensive. So that's what the DNA is. It's all the information. And that information is carried on from volume of print edition to print edition of the Encyclopedia Britannica. It tells you about, in a sense, what, in that case, the editors felt was the important information that needed to be known. Of course, in DNA, it's the all-important information that's needed to encode you, a rice plant, or a virus. Proteins are different. Proteins are the bits which are made from that information source. So it's kind of like a newspaper. It's the information which is printed on that day. It's important on that day where it's being actively used, and then it's torn up and thrown away, and the next day a new newspaper is produced. So it's much more short-term information. But that's actually also very useful. It's got much less information in it than a huge encyclopedia or a huge library, but it's telling you about what is happening then and there. And in that, there's enough information to answer some broad evolutionary questions. But for us as archaeologists, it's more interesting because it can tell us about the dynamics of what is going on in a particular context, in a particular sample. So I just want to also explain something which I think you all know, but I just want to make that point more clearly. DNA is much more useful for the main than proteins. And the reason for that is because only a very small amount of the genome encodes for proteins. We were very surprised when we finally sequenced the whole human genome 20 years ago, that actually there were very few genes, very few parts of the DNA that were encoding proteins. At the time we found about 20,000 genes, and now it's up to about 40,000, but the number of genes is much smaller. There's many fewer proteins being made, and most of the DNA is doing things we don't yet fully understand. So it's called non-coding DNA, but it carries lots of information about the copying process from generation to generation, and therefore lots of information that we can use to track migration, mobility, and identity. When you convert it into a protein, you actually make the information much more compact, and you're only using a small amount of the genomic information. So you're losing a lot of the power to do these kind of very high resolution inferences. I mean, your DNA is unique to you, and if you can sequence your DNA, without even seeing you, we could predict your eye color, your hair color, your sex, your skin tone, etc., etc. Proteins can't do that. It's this newspaper, it's this short-term information about what's happening in one particular tissue at one moment in time. We take the DNA sequence and we find what's called the start codon, which is this thing here. I'll go back again. So here's the start codon. We start reading here. And in fact, I've cheated slightly in this message. But then the message is read until we hit a stop codon, which is here. Actually, it's a bit cut off. There should be an extra letter there. So it's codified. So it's 3 letters for start, 2 letters for stop. And when we've done that, we can then read the protein. And these are the amino acids. Now notice that in the case of the DNA, there is only 4 bases, A, T, G, and C. But in the proteins, we actually have 20 different amino acids, and we have a selection of them here. And so we've gone from a very long and complex molecular message to a much shorter, more compact message. It actually takes about 1/6 of the atoms to write out the protein code than it did the DNA. So this more compact sequence is more likely to survive. And also, because we're made of 20 building blocks rather than 4, there's actually the information just about the composition of the building blocks, whereas the proportion of the amino acids can often identify the kind of protein. So there's even information in the broad composition. So here we've made our protein, but the important thing about proteins is they're not made simply by copying from the DNA. And this is why we misunderstood when we first found out there were so few genes encoding for proteins, because what happens to proteins after they have been made is they undergo a process of modification for function. So here our message doesn't read quite right. In this case, we need vitamin C, which is a vitamin found in fruits primarily. Most animals make vitamin C, but us and guinea pigs and bats, because we've been living on a frugivorous or fruit-bearing diet for so long, have stopped having the ability to make it. But what vitamin C does for us in these proteins is it converts an amino acid known as proline here, and we can convert it to a modified form called hydroxyproline. This is very important actually in our bones and our teeth. But we need vitamin C. If we don't have vitamin C, we can't make that conversion, and we get scurvy, a disease which makes us sick. It means that our wounds will reopen, our teeth will fall out, because we can't make the protein collagen properly. So once we add the vitamin C, we actually make the final conversion of the protein. So this is called a post-translational modification. We have translated from DNA to proteins. We're now going to further change it after it's been made, and we're going to get our final message. And now we can read what was encoded in the DNA. This is the protein product. So a much more condensed product, but one which is further modified in the cell. And actually then, because we have 20 different amino acids, all of which have different types of reactions available to them, will also show quite considerable modification over time. So what is proteomics? Well, this is what proteomics looked like 30 years ago. And this is how it came about. What you would do is you'd take a protein, a cell's mixture of proteins, and then you'd separate them out on gels, and you'd separate them out onto a clear gel made of polyacrylamide, first by charge here. So this is a positive end, so it's going to attract more negatively charged, more acidic proteins. Here's a negative end, so it's going to drag towards it more positively charged basic proteins. And then once we separate it out this way, we can lie that piece onto a second gel, which separates by size, with the larger proteins at the top and the smaller proteins at the bottom. And if you extract a cell, you get thousands of spots like this if you do a two-dimensional gel, first on charge and then on size. And what you would do is you'd maybe take two treatments. You'd take a cancerous cell and a non-cancerous cell. You'd extract all the proteins, and you get two sets of gels like this. And then what you'd do is you'd say, oh, what is the difference? And if you look very hard, you'll see that one of the spots has changed in position. So one of the spots changed position, and it was this spot here. And so that was what proteomics was. It was looking at spot differences. In fact, the zooms technology that we use comes from the fact that what people would then do is pick out one of those spots and try and characterize its fingerprint after you digested it with an enzyme. So it was a way of characterizing, fingerprinting these spots. And so that's a classic 1990s proteomics analysis. But the big breakthrough was the ability to get proteins into mass spectrometers. This is hugely powerful and important. And what it meant is once we got them into mass spectrometers, we could then break them apart and look at the characteristic masses. We know the mass of each individual amino acid. And from that, we could identify sequences. And then because we had these things called post-translational modifications, which also led to a change in mass, we could also identify those and assign them to particular places on the protein. And so there are two sorts of ways that we can do this. The zooms method is a very high throughput, quick fingerprinting method. It came out of that 1990s technology. You take a pure protein, it was normally a spot taken off a gel, you put it onto a metal plate, and what you then do is, having digested the protein into individual peptides, so the individual slices would cut usually at 2 of the 20 amino acids, the 2 basic ones called arginine and lysine. So typically that's 2 of 20, about 1 in every 10 amino acids would be one of those. You, on average, would cut peptides of about a length of 10. It varies from length of 2 or 3 to up to 30 or 40, but the average length is about 10. And because amino acids have an average mass of about 100, that meant on average these peptides were about a mass of 1,000. And that was good because the mass spectrometers can only detect over a range of between about 0 and 4,000. And most proteins have masses, and the average mass of a protein is about 60,000. So you can't measure the whole protein. You have to cut it into bits. Now, that's useful for us because in archaeology, the proteins get cut into bits by time. So a technology that actually works with bits and fragments is ideal for us. We're lucky that we're working with this kind of technology. And when we've done that, we can take those fragments and we look at them on an MS scan. This is from small to large here. And every different protein is made of a different set of amino acids. and the arginine and lysines, the points where they're cut, are in different places. So those fingerprints are pretty unique. So we can identify from the fingerprint what it is. And the Zooms methodology basically targets teeth, antler, bone, and ivory, mineralized skeletal material, where there's one dominant protein, collagen. So you don't need to pick a spot out of a gel. Archaeology, over the course of even just a few years, in fact, you tend to lose most of the other 600 proteins in a bone, and you're mainly left with pure collagen. And it's actually very easy then to purify the collagen out of the bone. And you've already heard about isotopic analysis, and a lot of isotopic analysis is actually done on bone and dentine collagen, pure collagen. So we take that same pure collagen, we can do a fingerprint, and from that we can identify what the species is, and that's what Zooms is. And this metal plate you can see here has got different spots, and on a typical metal plate you can spot 384 samples. So many of those are controls, but you can certainly analyze something like 100 samples in the course of an hour and identify all of these fragments. I'll talk a little bit about that later. So it's very quick, it's very cheap. On the other side, we have a much more expensive method. On the face of it, it looks very similar, but instead of having something on a metal plate, which easily goes into a mass spectrometer, mass spectrometers are in a vacuum, you have a liquid which gets injected into a mass spectrometer, and in the injection process, the solvent around the proteins is lost. Again, this has been fragmented and cut with enzymes. It then goes into an MS scan, so we can scan all of the masses, and then one particular mass is picked, and then that is further smashed into fragments itself. So it's an MS to the 2, this is the mass spec scan of 1, this is the first scan, and then the second scan, so it's an MS experiment. And here now we have actually fragments, and each of these different fragments is hopefully identifying a different length of that peptide as it's got broken to shorter and shorter pieces, and that is allowing us to work out what the sequence was. So this one can tell us what the sequence is without knowing it. This one we have to know we have a pure protein, and then we can tell what that protein was by comparing it against the database of other fingerprints of that protein. So this is good for fingerprinting against known. This methodology we use to find out unknown sequences. And it takes a lot longer. So this will take about an hour and a half to 2 hours from the experiment. And because these machines are very expensive and the analysis is complex and we have to have many, many blank controls in between, this analysis probably costs something like 50 times more than that analysis. So this is the example is to go through the LC-MS experiment. We take a protein mixture, we digest it, we then separate it, sometimes in 2 different dimensions, sometimes in 1. We generally just do 1 dimension. We separate it usually on how charged it is. We then fragment it. We find a mass we're interested in. We smash that mass up. And then the important thing, once we smash that mass up, we often talk about a thing called protein sequencing. Oh yes, we sequence an ancient protein. That is not a very good expression. When we do ancient DNA sequencing, we actually sequence the DNA. When you sequence an ancient protein, it is smashed to pieces, it's fragmented, but then to identify the sequence, we have to compare it with other database examples of other peptides. So it is theoretically possible just to guess or work out from the difference in these masses. That's quite a small mass, that one's quite a large one, that's a very large one, maybe that's 2 amino acids, that's a very small one, that's probably glycine, the smallest. And you can sort of work out the sequence based upon the fragmentation pattern. But actually what you normally do is you say, I know what that is. This is a sample of rice, or this is a sample of milk, or this is a sample of something. And in milk, we have these proteins. So we put a database together of all those proteins, work out what all of the peptides we can generate from those proteins are, and then we compare our spectra against a set of known spectra until we find the best match. So protein sequencing is actually about probability matching And of course, if you haven't got, and this is often the case in archaeology and even more so in paleontology, if you haven't got those sequences in your database, then you will not find the protein. But annoyingly, the software will be so helpful, it will suggest the nearest match in the database, which will probably be the wrong thing, or necessarily will be the wrong thing, because you haven't got that sequence in your database. And so one of the things in proteomics is to work out how confident are you really that you have that particular sequence. Okay, so that was my introduction to proteomics. Now what I want to do is think about it much more in an archaeological concept. And this actually comes from work by Marcos Martonian Torres, who's at Cambridge, and he's a metallurgist. And he's interested in object biographies, so the biography of artifacts. But if you think about a living thing, and I've done a shell here, I've chosen a shell, then as an archaeologist, and we see something like that on the site, we say, what is it? What species is it? Where did it come from? So what is it? Identity is the most important thing. And then we say, how old is it? You know, what is the age of the sample? Is it in context, or is this something which has actually been later introduced to the site? We want to say, well, where would it have come from? You know, is this something which has come locally to the site, or is this something that's come from distance? And we might want to say, if it was a human bone, let's say, what is its ancestry? I mean, maybe even in this case, this is a shell, but we might want to say, well, we know that this is a recently derived species, which is coming from a particular lake. What is the ancestry of this group of shells 1,000 years ago, 2,000 years ago? And then we want to know about the history of the object itself, like how old is it? This is a young animal, is it collected in the spring or the autumn? And then we obviously worry about, well, we found this shell, but this is a very robust type of shell. Maybe they were collecting much finer shells as well, which got broken down, or the way that they cooked or processed the samples. We worry about taphonomy. So we take a living thing, and it could be, we could have taught that about human skeletons or a shell, whatever you like. We're interested in a set of questions. And what Marcos has pointed out is if we take an artifact, a produced thing, and he works on metals, so he's caring about those. But again, we work on organic molecules because, or we work on material culture made from living things, because proteomics, that's where the things are coming from. Again, we want to know the date of the date, and it's rather than location, we want the provenance. And rather than ancestry, we want to know the manufacturing history, the style of manufacture. In terms of biography, we want to understand the use history of the object, and again, we want to look at taphonomy. So what I wanted to do is think about that kind of cycle through what proteomics can do, both for the living thing and for the artifact. So in terms of identity, and the reason I actually chose a shell is because this paper has just been published in eLife, and they are looking at the use of mother of pearl in prehistory. And what they are doing is now using proteomics, because these shells, this is a team based out of Turin, because shells have proteins in them. And so if you're looking at mother of pearl, which is obviously widely used, and because it's highly processed, it's very hard to identify what that mother of pearl is. Well, it came from a mollusk. So if you look at the proteins in the mollusk, you can identify different species of mother of pearl. So we can do identity with proteomics, and of course we can also do that with DNA. The next thing we can do is, I actually missed out one I should have put in, date. Because the proteins themselves decompose, one of the things that I'm extremely interested in is looking at the way that the proteins are decomposing for dating, and we have a technology of racemization dating for dating. Location problems we can't do. Proteins are not very rapidly evolving. As I mentioned previously, DNA has a much richer source of information. And if you take the same, you'll be very, very unlikely to find any protein difference. There are some that are so few I've left it out. We can't do location. We can do ancestry though. And here's an example, a Chinese example. I think this is work that's come out of our lab in Copenhagen, led by Frida Welker. And he's looked at this extraordinary creature, which we know primarily from its teeth, which is a thing called Gigantopithecus, this huge ape, And I love the fact that here's two different illustrations of it. This whole thing is drawn just from these teeth. It's because the porcupines eat the rest of the skeleton and they only seem to leave the teeth behind. This comes from Chufeng in southern China. And now what's extraordinary about this is a species that died 1.9 million years ago, the sample is derived from. And if we're trying to do DNA in a site in southern China, 1.9 million, you would forget it, you wouldn't even try to do it. But in fact, the proteins are so well preserved, we can put this species into the tree. We can see what it's most closely related to, and it's most closely related to Pongo, the orangutan. And we can also then work out what sex each of the teeth are, because we can do the sexing. But our ancestry is a very powerful technology if you want to look in very deep time. For proteins, if you want to look at the relationship between, say, two different races of horses, we wouldn't be able to do it. As I mentioned, that the resolution is not there. You have to use DNA. But if you want to ask big, bold questions of phylogeny, then it is useful. And in fact, what's good is that the proteomics also agrees with the very recent morphological identifications, this being most closely related to orangutan. What about biography? Well, that's another interesting thing about proteins, because... Sorry?
Don't worry, that was an accident.
Okay, no problem.
Yeah.
Because proteins are being expressed, and the expression of proteins will actually change during life. So here's a nice example of that, maybe one you haven't thought about. But this is an example also from the team in Copenhagen, where they were finding very well preserved leather artifacts. These were actually cloaks, leather artifacts in the bogs in Denmark. And they wanted to ask the question, how high quality was the leather? And so what they did is, by analyzing the leather using the complex, expensive proteomics technique, leather is mainly collagen, but they actually found in the leather calf fetal hemoglobin. And as its name suggests, it's produced when the calf is in utero, in the fetus. And what it is because the calf is trying to get oxygen from the mother, it has to have a form of hemoglobin which is better at dragging oxygen away from its mother. And calf fetal hemoglobin is no longer produced by the age of about 6 weeks. This leather had calf fetal hemoglobin in it, and what that tells you is that this must have been calf skin leather, so very fancy leather made of calf skin. So we can get the age of the skin, and from the age of the skin, we can get information about the choices that are being made about that object. And then the other thing about, I guess, biography or identity, and this has been exciting work over the last couple of years, and something which is going to certainly grow and grow, is that strangely, the main protein in tooth enamel is coded both on the X and the Y chromosome. So you actually have differences in the sequence on the X and Y. And when you analyze the proteins in tooth enamel, if you have a male, you see additional peptides, which are peptides produced just by the Y. You can see an example. This is a Y-specific peptide. And so this is an intriguing example, because this was a famous burial of two lovers, the lovers of Medina. And then when they actually, the DNA preservation wasn't good enough to get a DNA sexing identification, but you can sex these with enamel, and actually the lovers of Medina are both male. And you saw that also in the Gigantopithecus, and there's been a recent paper looking at Homo erectus, and in the Homo erectus sequence there was insufficient protein to say anything about its relationship to other hominins, because there wasn't enough protein sequence to give a good detailed phylogeny. And remember, proteins are a crude way to get phylogeny as an information tool, but it was good enough to sex. And so I imagine moving forward, if you think about all the questions in zooarchaeology, and we're currently, for instance, working on sheep and goat sexing, because we're interested in animal husbandry. So there's lots and lots of questions where you'd like to get sex. And if you have a methodology which is so robust, it survives for millions of years, even in the hottest archaeological site you're going to work on, one with the worst fossil preservation. As you probably know, when you get poor preservation of skeletons, you lose the bones first and the enamel last. So you're almost certainly going to be able to do sexing using proteins. And then the last thing which is kind of interesting here, I think, is about taphonomy. And this is an example from Frida Welker, who's in the group in Copenhagen. And he was asking a question, when you actually look at these animal remains, you're identifying these remains, is there any difference perhaps between the remains that we can identify zooarchaeologically, because they're large enough, we have enough features to put them into species, and the fragments which are so poorly preserved that we can't see them? So here he looked at a cave site, and in this cave site we have got morphology, and in morphology you can see that the archaeologists were separating them out in size terms. We see a lot of cervids and caprines in terms of the size of the individuals, and we have a few bison and a few horses. But however, if you take the exact same sediment and you look at just the tiny bone fragments, which are far too small to be identified zooarchaeologically, what you see is actually there's far more bison. I mean, cervids are the same, really, far more bison. And what they interpret in this paper is that the bison have been very heavily exploited on this site, so much so there is almost no large bone fragments left. They have been broken up, they've been extracted for their marrow, for their fat, they've been destroyed. So just by looking at the morphological record, you may miss out some of the total record, and therefore potentially proteomics is useful taphonomically, particularly when it's so cheap, to this very, very simple Zooms fingerprinting. This is a Zoom study. Okay, so that was the living thing. What about the artifact itself? We have made something from a living thing. We want to ask questions about the artifact. Now remember that in a living organism, the vast majority of the organic matter, most things are water primarily, famously, you know, the Archbishop in the Church of England is 60% water. I mean, everything is water. We are bags of water. But taking the water away, then we are 50% pretty much protein, ignoring us in the organic part, ignoring our skeleton. So proteins are everywhere. Our hair is protein, our muscles are protein, our skin is protein, our blood is protein, our milk is protein, you know, it's all protein. And therefore, when we make things out of living things, plants or animals, we will make quite a lot of those out of protein, and we can see that in the artifact. So we can detect the protein in the artifact. So the first thing we can do is identity, and this is an example which I thought was rather fun, which is where we took bone points. And these were bone points that we found from a site in Canada. And what was most interesting, I guess, about this was that we were able to identify the species just by analyzing the bags that the samples were stored in. And this was a site where, on the site, it was an Iroquois site. It's historical, so it's from the 16th, 17th century, so it's quite recent. The preservation was good, but on the site it was mainly fish bone remains and some white-tailed deer. And we assume that all of the artifacts would be made of white-tailed deer. There are some, indeed, artifacts made of white-tailed deer. Some of the artifacts were heavily burnt, you can see here, and when they're burnt, there's no proteins left. But what was a real surprise to us was the number of artifacts that were made of either bear or human, because whilst there were humans obviously active at the site, because we could see the activity, there was no human remains, but the humans were active at the site, there was no bear bones at the site. So, but in fact, these artifacts were made of human and bear, and the archaeologists that were digging the site were extremely happy with what for us was a very puzzling response, because in the tradition of the Iroquois, there was a whole human-bear transformation process, and so they saw these hunting artifacts as actually linked to that tradition of human-bear shape-shifting, And for instance, the idea that if a family is starving, the youngest child would turn themselves into a bear, go into the forest, fatten themselves up, so the family could later go back and hunt the fattened bear who'd collected the goodness out of the forest, and they could eat the bear, eat their youngest child, who was feeding the rest of the family. So for them, the identification was important. For us, it was useful because we could analyze these artifacts without destroying them. And then there comes a very interesting ethical question once you discover you're analyzing human remains on an archaeological site where you didn't expect to find human remains. And that was a point where the archaeologists went to the tribal groups in the local region and said to them, would you want to study these further? One of the reasons was because the date was such that it was just at the point of contact between the Iroquois and European settlers into North America. And one of the questions was, were the human bone points made of essentially European bone? And it turned out that none of them were. But that was one of the questions they wanted to ask. We actually then, in some cases, did do destructive analysis with DNA because you can't do that from protein. So we can look at identity. Provenance is something else that we can do. Again, we can do this from an artifact basis because whilst we can't, there's not enough sequence variation to say where a particular species comes from based upon its protein sequence. Because different resources are geographically separated, by identifying what a resource is, we can say something about where it has come from. And so this is an example where we were analyzing books. These are this huge tome here. This is a book which comes from a very important Cistercian monastery in the place where the Cistercians founded themselves. So this is a very important collection. This is in France, and a very important collection in France. And the team noticed that these books had fur on them. And so this fur had been puzzling researchers for some considerable time. And it had originally been identified as pig or cow. But then, looking more closely, it was identified as being made of deer skin. And in fact, we have analyzed now proteomically a number of leather bindings from this period and earlier. And we found quite a few of these which are on deer skin. But when we analyzed, including up until the 11th century, but when we analyzed these books, they were actually on seal skin. And that's curious for a number of reasons. At this period of time, there was no word in French for seal skin. And in fact, if you look at pictures of seals, these marine mammals painted in these manuscripts, they clearly have never seen seal. And we were able to identify one of the seal skins was a species of seal which is only found in the Arctic. So what that told us was that at least some of these seal skins must be coming from the Arctic. And at the time when this book was made, that would mean that they almost certainly came from Greenland. So the proteins themselves can't actually say it's from Greenland, but by identifying the source, and if we know that the source is patchy geographically at this time period, we were able to do that. In fact, in this case, we then cheated and we went to DNA and we sequenced not only the Greenland seal, but also some more common seals which were found around France. And it turns out that even those seals, they all came from high Arctic sites. All of this was seal skin which was being traded almost certainly by the Vikings down into France at this period of time. So we can get ideas of location. Manufacturing is another thing where proteins, because of the way that you manufacture and process a sample, you can actually learn something about the history of production. And this is a Chinese example. It's not my example, but it's one of my absolute favorite examples, because I think this shows the power of proteomics. And this was this site. I daren't pronounce it because I'll pronounce it incorrectly. But at this site, there was evidence of cheese. And because the site is very dry, if you can see around here, there's these kind of strange lumps of cheese making almost what looks like a necklace around the mummified remains. And you can see from the fabulous preservation just how in these very dry conditions there's excellent preservation. And in this project, the team from China and Germany analyzed these proteins, and they could see a range of things. But to me, the most interesting thing is, first of all, when you look at that material, you can tell it's cheese because cheese is derived from milk. But in milk, you have two major types of proteins. You have the casein proteins and the lactoglobulins. The lactoglobulins are soluble and are found in what we call the whey. The curd proteins, the caseins, are acid insoluble, and they are often used to form things like cheeses. And this was much enriched in the cheese proteins relative to the whey protein. So it was a cheese. And that's kind of interesting. So the cheese part of milk, not the whey part. But to me, the most elegant part of this was that when you make cheese, you can do it in many different ways. To precipitate the curd proteins out, the most common method of doing this was to use acid. So if you acidify, and you can do this by fermentation, there's various ways you can acidify the protein, and the curd proteins will precipitate and fall out of solution, so we've separated out an insoluble curd from a soluble whey. But sometime in the distant past, someone who must have been hacking at a young calf or a young sheep, when they took out the stomach contents, they noticed that in the stomach, the milk was all in the form of lumps of curd and soluble whey. And they must have thought, well, if we put, or maybe if we put more milk into that stomach, we might, into the intestines, we might actually use it as a way of carrying the milk. And what they discovered was that actually in the digestive tract of young animals, there are enzymes which will cut at this protein, but at one specific site. And this idea of what's now called the use of rennet, these enzymes which are very specific, it's an enzyme called chymosin or renin, but it clears at one position, and the position is a very strange position between this, and there's 20 amino acids. It's between the F, phenylalanine, and an M and methionine, so it cuts here. As I mentioned previously, normally in proteomics, we cut with trypsin, and it cuts at arginine R or lysine K. But this cuts between the F and the M, which is very curious. So when this team then took some feta cheese, so some good Greek feta, feta is used making rennet, and you can see that the peptide stops at the letter F because it's been cut with rennet, and that's where rennet cuts. When they looked at this cheese, it went right through the F, and it stopped at the next triptych cut, which is the letter K. And what that's telling them is that in this particular case, the cheese was made by acid precipitation and not the use of rennet. So it's a very simple observation, which is extraordinarily powerful, because what this now means is we have a proteomics technology which we can use to test for the idea of the use and expansion of rennet. And we have the use of rennet is described in cheese making for the last 2,000 years, but we don't know how much further back in time it went. But with proteomics, we can now tell. So we can look at manufacturing. We can then also look at use history. And this, I think, is one of the most exciting areas that is now available to us with proteomics. So this is work by Jessica Hendy, who's at University of York. And she's been looking at the site of Çatalhöyük in Turkey. And what Jessie was wondering, so Jessie and I have spent a long time working on dental calculus. This is the calcified precipitation of the pellicle in the teeth and looking at the proteins in dental calculus. But Jessie was discussing with some German researchers who'd been working on the East Mound, the most recent part of this very, very early settlement site in Turkey. And they had got limescale on their pots. And Jesse was wondered whether or not the limescale could be treated like dental calculus, and could we get proteins from it. In the site, you can see it's dominated by sheep over goat. What Jesse was able to do was to extract both from the ceramic matrix and also from the limescale, but you'll see the limescale is much richer. Looking at the ceramic matrix to begin with, you can see we have here the indication of what she found. So blue indicates dairy, and if it's a solid symbol, it indicates we could actually tell what the species was. So we have definite instances of goat and sheep dairy here, and we have instances of sheep meat or blood, because it's a blood protein we're finding, which could be found in bone. Here we're finding what we call bovidae. What that means is we found only those peptides which can say, yes, this is a milk protein, but not the peptides which say which species of milk it is. So if you've got a poor preservation, you may not get those peptides which are different between the different species and can uniquely identify them. But you can see on the whole, there's not a huge amount, and we have here some plant protein. Again, preservation is not good enough to say what kind of plant, but we have a sequence which is from plant. So that is actually looking at the non-limescale component. However, when Jesse looked at the limescale, it's a sort of very fine formation of lime, of calcium carbonate on the surface. And this is something which will be very amenable to Chinese research, because there are many, many sites in China which have got heavy water, hard water, which have got, therefore, the formation of limescale on some of their deposits. And as you probably know, the tradition has been, particularly in Mediterranean archaeology, when you get limescale on ceramics, the first thing you do is dip it in acetic acid so you can get rid of the limescale, so you can see the fabric of the ceramic. We're saying, no, don't dissolve away the limescale, just let's dissolve it away under controlled lab conditions and get out the proteins from it. Once you did that, you can see a much, much richer story, so that there's a much better preservation of proteins in the limescale than there is in the ceramic. And what you can see here now is, first of all, you can see again dairy and meat. But what's much more exciting for me about this study is we're beginning to see many more plants. So we can see a range of plants being combined together with milk and meat in different vessel forms. So this is, one of these questions we begin to actually start to open up the whole question of how we're beginning to process foodstuffs in different ceramics. And you can see why I think many of you might be excited about this, because the potential to begin to start to unlock the different uses of different materials, and also to be able to compare the archaeobotanical and archaeological records on the site with direct evidence from different ceramic vessels at different periods of time throughout a sequence could be very powerful. I'm going to give you a bit of a downer on that at the very end, because our new results are not as good as those, but I'll show you those at the end. But it's an exciting, really, Chattal has given an amazing result. And so, for me, this is one of the interesting ways in which we want to begin to bring back together the kind of traditional science work which has been done by archaeological scientists and archaeologists. And so thinking about a concept of object biographies, thinking about the way that objects have passed through the life cycle in the way that I've shown you in the figure which I've drawn from Marcus. We have to be careful, I have to say, about this, because it's very, very exciting to get lots of proteomics data. But one has to be careful about the quality of the results we get. And the reason for that is because we are interfacing with proteins all the time. And in any biochemistry lab, they're using many, many proteins. And so this It was a report from an Iron Age site, where on this Iron Age site, they found peptides from a Crimean Congo hemorrhagic fever virus, so very curious virus, which is mainly found in Africa and very southern Europe. In fact, in Europe, it's only found in Sicily. It doesn't, the creature that carries it doesn't live any further north than that. And yet this was a site in central Germany. It's not a conditionally defined sample. And it's so it seemed very strange. They also found cat blood and human blood and all kinds of strange things. In a review we wrote recently, we made this point, which was that you have to look at where that sample was analyzed. It was analyzed in a lab where they actually specialize in the study of Crimean Congo hemorrhagic virus. And worse than that, the actual peptides that they found are used in that lab to make synthetic vectors. They're making huge quantities of this particular protein in that lab. So we almost certainly think that was contamination, just because if they'd done the same analysis of these pots in any other lab, they would not have found that virus. And the problem comes if you start to look at all the proteins that we care about. We're interested in milk, we're interested in wool and hair, we're interested in glues and skins, we're interested in eggs, we're interested in maybe rubber, I suppose, we're interested in muscle tissue, we're interested in lots of disease species, we're interested in cartilage type materials and skin. Many of the proteins that we care about are extremely common laboratory contaminants. And so many a time we've been fooled into thinking we have a very exciting result, when on more careful analysis, we realize that it's actually an introduced contaminant. And so finally, we want to worry about taphonomy. So one of the things we're trying to show in terms of, is this real or is this ancient, is actually looking at the damage patterns and the patterns of survival. And you can see here, this is actually dental calculus. But this is just showing you, this is very crudely separated in time, that we are seeing less proteins in old samples. That's not too surprising. And proportionately, for instance, we only find plant proteins in modern samples. So there is definitely some kind of taphonomic story, and if you found very exceptional preservation in a very old sample, you begin to worry about it, and we've actually been trying to do something about that ourselves. And so in our review, we wrote a review in 2018 where we try and sort of guide you through, if you're doing some of these experiments, the kind of things you should be thinking about, how you've got the original signal, then you have microbial growth, there's contamination after excavation, after extraction, and then in the machines that you do the analysis. So that you have to be extremely careful looking at your data to see how diverse the signal is and how meaningful the signal is. And so the next two slides just come from that paper. But if you look at its Guide to Protein Studies, published in Nature, Ecology, Evolution, these are the things. I won't go through them, we haven't got time, just to think of if you're doing those kind of things. I'll very briefly stop on future directions, and I want to say one thing. One thing we are trying to do as a community at the moment is we use this Zooms technology, and we had a meeting in Copenhagen 2 years ago, and that is now moving on to trying to put together a set of online protocols and freely available software, because we want everyone to be able to do this analysis. It's very cheap, it's very simple. Undergraduate students can be trained to do this in a day. It's not hard. And the only hard part is finding the mass spectrometers, but to be honest, they're sufficiently frequent. We generally just prepare stuff in Copenhagen, just post it to someone else's mass spectrometer. So one thing we're trying to do is to make Zooms more democratic. Another thing we're trying to do is, so while proteins are great on their own, but what we've learned particularly from the work on dental calculus is what you should be doing is looking at everything together. So you look at the starch grains and the phytoliths trapped inside, look at the metabolomic signals, you look at the DNA, the proteins, look at the isotopes, look at the evidence of things like lapis lazuli, and then you link it to the individual's age and life history. So you don't just do proteins on its own, do proteins as part of a larger integrated study. And the other thing that we're kind of very interested in now is we're very upset by the way that particularly human remains are being destroyed to try and get things out of them. And so I'm very interested in the work that's just been published, or last year been published by Matt Sponheimer, on using screening methods which actually can detect the presence of protein. So this is using near-infrared to look at collagen levels in bone. And so we've been using this as a method with Matt to try and screen bone so that we don't destroy samples where there is no protein there. Finally, sort of about what we're going to be doing in the next while. We've just started a project, a new PhD student, Ali Morton-Hayward at Cambridge. So she's been discovering that there's huge numbers of ancient brains preserved. And so she's been analyzing the preservation of bone proteins, and these are just two ancient brains. This is the Iceman and Hesington brain. This is looking at, in blue, the parts, the sequence that we're recovering. And in fact, the Tyrolean brain, the Iceman's brain, has better coverage of this particular membrane protein than any modern brain protein study we've seen up to date. So the preservation of ancient brains is amazing, and there's enormous numbers of ancient brains around. So if you're interested in working on ancient brains, please get in contact. And then the other one is Miranda Evans. So she's coming with Jesse and ourselves at Cambridge. And so she's now looking at Roman pottery, and we're doing a project on Roman pottery. And as I said, I'm kind of disappointed about this. So here's the limescale results which she's just been getting. The numbers are very small. But what we're seeing is, see here? The very small amounts of plant protein we're getting. And we were very delighted to see lots of plant protein in the Chattel dental calculus sites. This is now looking in Roman ware, mortaria and things. In the Roman ware, we're not seeing so much. Is it to do with the use of the pots, or is it to do with the fact that it's not as warm, so we're not getting so much limescale forming, so we're not trapping as many plants? We don't know at the moment. So one of the things Amanda will be doing is doing some experiments to try and simulate the process of incorporation of foodstuffs into limescale. Okay, that ends my hour. I think I'm almost to the dot.